Hi there, Dr. A again. So let's do bacteria part 2. And we're gonna look at nutrition and metabolism for bacteria, but we obviously are gonna start with a case. So this case is a 22-year-old woman that presents at the walk-in clinic with a slight fever, which is 38.5 Celsius, a complaint of frequent urination, burning on urination, vaginal discharge, and a small lesion on her labia. The woman reported that she was moderately sexually active and had 3 sexual partners in the past 6 months. Her last sexual contacts were about 7 days earlier. She had developed mild symptoms about 5 days earlier, beginning with a discharge from the vagina. She began having pain on urination about 3 days earlier. Urine analysis revealed a pH of 8.2, which is alkaline, some white cells, and a few red blood cells. There was protein in the urine. A smear of the vaginal secretion showed a number of gram-negative cocci. Okay, so gram-negative cocci, vaginal issues. So which one do you think this is? So obviously everything that I've listed here can occur in the vagina. So we have Candida albicans, which is a yeast, Trichomonas vaginalis, which is a parasite, Group B strep, which is bacteria, Neisseria gonorrhea, which is bacteria, and Chlamydia trachomatis, which is bacteria. So which do you think it is? I probably just eliminated 2 of them just by reading them. So give me a guess of what you think. Okay, and if you guessed it right, you guessed Neisseria gonorrhea. She has gonorrhea. So gonorrhea is a gram-negative bacteria. It's a cocci. And when we get the smear from the vaginal sample or penis sample, you'll get a lot of white cells, and the bacteria are actually inside the white cells because the white cells have eaten them. So, and you see them occurring in pairs here. So they're a diplococci. They tend to occur in pairs. This one's a little bit harder to see. The bacteria is a lot smaller, but you see a lot of white cells with intracellular gram-negative cocci. And Neisseria gonorrhea is grown on a chocolate agar plate because it's very fastidious. It's hard to grow, and this plate's called Thayer-Martin plate. And Neisseria gonorrhea is what can grow on it. And so the colonies look like this on the agar plate. Okay, so also known as the clap, yay. So gonorrhea spreads easily and can lead to infertility in both men and women if untreated. Antibiotics stop the infection. Common symptoms are burning during urination and discharge, but often there are no early symptoms. Later, the infection may cause skin rashes or spread to the joints and blood, so that's not good either. In men, you see this discharge from the penis and swollen testicles, and in women, vaginal discharge, pelvic pain, spotting. Symptoms may be mild and are easily confused with a urinary tract or vaginal infection. Okay, so treatment can be sometimes a little bit complicated. It's like it needs a couple of things to treat it. So if you have uncomplicated gonorrheal infection of the cervix, usually they do cefixime, and if chlamydial infection is not ruled out, because sometimes you can have co-infections, you do azithromycin also. So you can do basically any of these. So Cipro and Z-Pack, Zithromax type stuff. And if it's in the pharynx, y'all can imagine how that happens. Single dose, again, of the Cipro type stuff or ceftriaxone, and some Z-Pack also. So we're going to talk a little bit more later on this summer about antibiotic treatments and stuff. I just happen to have that one on there. So this one is important for it to be treated correctly because it is a bacterial infection, and it does need to be treated with antibiotics, but the proper course of antibiotics, of course. So let's talk about bacteria and what their chemical compositions are, and that's going to tie into their nutrition. So Neisseria is a fastidious one, so it has high nutrition requirements, but really, like, what are their nutritional requirements? So a bacteria is made of proteins, nucleic acids, so your DNA and RNA, carbohydrates, lipids, and then some other miscellaneous molecules and stuff, which we're going to look at. So if you look, carbon, oxygen, and nitrogen and hydrogen are all the building blocks of these guys right here, of the proteins, nucleic acid, carbohydrates, and lipids. Your carbohydrates and proteins just don't have nitrogen. And then your DNA and RNA also has phosphorus. Some of your proteins may have phosphorus. Some of your proteins may have sulfur. And then some of the miscellaneous could have been your potassium, sodium, calcium, magnesium, chloride, iron, and your trace metals and stuff. And of course, it is also composed of water. This is percent dry weight. So you can see it's predominantly these building blocks right here, which are these atoms right here, these elements, which are the main components of this. So they're made up of the same kind of stuff we are. Our cells are made of all of this stuff too. So very similar in composition. All things that are alive really have carbon, hydrogen, nitrogen, oxygen, and stuff as building blocks. So it's worth here talking about nutritional categories of microbes. So by energy and carbon sources, there's two things. To stay alive or to be alive, you have to be able to produce energy so that you can drive chemical reactions and stuff like that. Bacteria too. And they need a carbon source because carbon is the backbone of carbs and lipids and nucleic acids and all that kind of stuff. Okay, so we have basically categorize them on how they provide these two things. So we have autotrophs, which are troph is like think feeding. So autotroph are like self-feedings. And heterotroph, hetero is different. They have to get their sources, their feeding outside of themselves. Okay. So in the autotrophs, you have photoautotrophs. So their energy source, and they're in the non-living environments. And so their energy source is sunlight, and their carbon source is CO2. And these are your photosynthetic organisms such as algae, plants, and cyanobacteria and stuff, in oceans and lakes and all of that. And then your chemoautotrophs, their energy source is simple or inorganic. So like soil and different stuff like that, or rocks. So it's and their carbon source is CO2. Only certain bacteria, such as methanogens and deep sea vent bacteria. So these are kind of unusual little guys right there. And then the majority of your bacteria are going to be in this category here. So heterotrophs, there we have photoheterotrophs. So they use sunlight as their energy source, and other organisms. And their carbon source is organic. So that means that from other living things and all that. So you have purple and green bacteria, photosynthetic bacteria, are here in your photoheterotrophs. And then the majority of our pathogens and stuff like that is going to be here, or chemoheterotrophs. So they do, for their energy source, metabolic conversion of nutrients from other organisms, including ourselves and stuff. Their carbon source is organic, again, so other organisms and us. So these are your protozoa, your fungi, many bacteria, and animals and stuff. So we are chemoheterotrophs also, because we get our energy source and our carbon source from other organisms, with a plant, animal, and all of that. And those are organic, life-based forms and stuff. Within your chemoheterotrophs, again, is going to be in a majority of things. We are, most of the bacteria, pathogenic bacteria, good bacteria, all of them are chemoheterotrophs. You have also subcategories. You have saprobes and parasites. So saprobes help you metabolize the organic matter of dead organisms. That's their source of energy. And so these are fungi and bacteria that are decomposers. They're the ones that break down dead bodies, but also just any kind of dead organic material and stuff. And then parasites, they utilize the tissues and fluids of their host, but of course the problem is they do cause damage to the host. So we have Various parasites and pathogens would fall under that. They can be bacteria, fungi, protozoans, or animals here. And so I know we're kind of focusing on bacteria, but this is just nutrition, microbial nutrition in general. Okay, so a couple questions on this quiz. So select the molecules that are found in bacteria. So we went over some of those. So what molecules? And this is, you can select multiple of these, okay? And then your second question is, which carbon and energy source would Neisseria use? So the bacteria from our case study, which one do you think it is? And just select one of them. Okay, and so now let's think about, okay, so we know bacteria have and other microbes have a requirement for carbons and hydrogens and nitrogens and all of these things. They can get the majority of them. The heterotrophs will get their source from living things, but bacteria don't have mouths. Like, how do they eat? How does this stuff come in? Not like us, right? We're a complex system. They're unicellular. So how do they get the nutrients into cells? So we're going to look at how stuff moves in and out of cell. First one is osmosis. So osmosis is the movement of water from an area of higher concentration of water to lower concentration of water. And so if you take a bacteria and we're going to talk about put it in an isotonic solution, it means that the concentration, if you will, the solute concentration on both sides of the membrane of the bacteria is the same. So water will actually flow back and forth in and out of the cell. And so this is showing for a bacteria, and then that's showing for like a eukaryote, so one of our body cells. So there's always this movement back and forth across the membrane, but there's as much going in as going out as the movement of the water moves, because the concentration of water is the same on both sides of the membrane. Okay, then you have it if you put it in a hypotonic solution. So hypotonic solution would be like pure water where there's like no solutes. And so you have a high concentration of water in a hypotonic solution on the outside of the bacteria or the outside of the cell. And there's more water on the outside of the cell than there is on the inside. And the inside of the cell is more concentrated with solutes and stuff like that. So what happens is then water is going to move in. This is going to try to equilibrate the concentration, solid concentration on both sides of the membrane. So water will run in, and for bacterial cause the cells to swell, and for but usually not always burst because of the cell wall. But our cells, and eukaryotic cells that do not have a cell wall, the water could rush in to the point where the cell bursts. If you put them in a hypertonic environment, the solute concentration is stronger on the outside. So basically there's more water on the inside of the cell than there is on the outside of the cell. And basically, so that would be like really salty water. If you put a bacteria or eukaryotic cell in that kind of an environment, the water will leave the cell, will be pulled out to the environment, trying to equilibrate again the solute concentration on both sides by moving water, and the cell will dehydrate. And because it will lose all its water, it will shrink and dehydrate. So, so that is osmosis, so the movement of water in and out of cells via the membranes. Okay, so now that was osmosis. Osmosis was just a movement of water across the membrane. But there is also diffusion. And so diffusion, simple diffusion is just the movement of molecules from a higher concentration to a lower concentration. And so this can be from anything. So this one shows like a sugar cube. And so if you put a sugar cube and you drop it in water, the sugar molecules are going to come off the cube and start spreading throughout the water and they'll move around without you even stirring it until they are completely evenly distributed across the water. And so so movements can happen, molecules are moved by diffusion, simple from the presence of going from high concentration to low concentration. So one molecule that will move by simple diffusion is oxygen. So it'll move from high concentration of oxygen to low concentration of oxygen. And so, or carbon dioxide, since there's a requirement also for certain bacteria, so it can move from high concentration to low concentration across the cell membrane. Facilitated diffusion is just a case of diffusion where you have to have a certain transport channel to allow that molecule to move through, but it always moves from high concentration to low concentration. So that's the thing of diffusion. And if you will, osmosis is a specific case of diffusion, and even facilitated diffusion, because water has to move through a transport port through the cell membrane. But it's the movement of water specifically, whereas facilitated diffusion can just refer to like any kind of molecule, high concentration to low concentration through a transport a protein port in a cell membrane. And then active transport, or carrier-mediated active transport, is usually a port here embedded in the cell membrane, and it requires ATP. So the molecule seeking entrance in there has to bind, and then ATP has to cleave and cause a conformational change in the shape of the port that allows that molecule to move from the outside to the inside. Now, this is active because it requires ATP, whereas diffusions, simple diffusion, facilitated diffusion, and osmosis are all passive transport because they do not require ATP. And the fact that this one, the body or the cell, the bacteria, the microbe, is willing to expand ATP to move that molecule, it usually moves against concentration gradients. So it allows to, even if there's a low concentration, for those molecules to move in and bring even more inside the cell. So a lot of nutrients are brought in that way because it's worth spending the ATP to bring them in because the cell needs it. And then there are a few others to mention. There's endocytosis, phagocytosis, and pinocytosis. So endocytosis, the cell encloses a substance in its membrane. So think about cells, think even like specifically like amoebas and all that. They do endocytosis. So they don't have a cell wall, so they're kind of free form and moving around. And basically they can wrap their cell membrane around an organism and bring it into a vacuole. And engulfs it, and then they can digest it using enzymes and stuff. So endocytosis is the process of bringing something into the cell by using the cell membrane to wrap around it and bring it into a vacuole. Phagocytosis is a form of endocytosis, and it's specifically accomplished by amoebas and white cells, and they ingest whole cells or larger solid matter. So endocytosis could be just for example, did you know you transport cholesterol into your cells via endocytosis? So cholesterol is a molecule, so that molecule docks on the receptor, and you actually just your cell actually brings it in by pulling it in through the cell membrane. So you can do endocytosis would be more like so molecules binding and being brought in, whereas phagocytosis is like it's bigger. It's a bigger form of endocytosis. It actually brings a whole cell or a solid matter, like a bigger chunk of stuff in. Okay, and then pinocytosis is endocytosis of liquids, so it's drinking or oils, so thorough liquid, anything in solution that is brought in. Okay, so another open-ended question. List one way chemicals can enter or exit a cell, just such as a bacteria. So you can go ahead and enter an answer there. Okay, and so another thing that's worth considering are the temperature requirements for the bacteria. So in the bacteria and just microbial world, whether we're talking about bacteria, fungi, parasites, and stuff like that, there are different requirements. So these here are called saccharophile. They really like cold temperatures. So we're going here is 0, that's freezing. This is in Celsius. So underneath 10, so like here's about refrigerator temperature. And so these guys can thrive at refrigerator temperatures and stuff like that. There are a couple pathogens like Listeria that can actually do really well refrigerated, and they can grow in the refrigerator and stuff. Your Sacrotrophs, they do like it, but they like it a little bit warmer than your Sacrophiles. So, these guys here, again, they can grow at refrigerator temperature, but they just really like room temperature. So about 20 low 20s to 24 degrees Celsius is a good room temperature. So they really like room temperature. So your Sacrotrophs like room temperature growth, and then your Mesophiles, they like body temperature. So about 37 Celsius right here is optimal temperature for these guys right here. So they can grow at room temperature. They can grow if you up the temperature. They don't like if it gets too hot though. So they do ideally to do best at body temperature. So all your pathogens and stuff are going to be here, and bacteria that colonize our body are going to be here. Okay, and then we have thermophiles. They really like hot. So this is way hotter than our body can be. So these would be some of the guys that like to, live by hot vents in the sea and stuff like that. And then you have even some that are extreme thermophiles that can thrive at really, this is beyond boiling temperatures and stuff. So kind of crazy there. Okay, so next is your poll. What would Neisseria be classified as? So Neisseria was our pathogen from our case. Was it a Sacrophile, Mesophile, or Thermophile? And let's look also at oxygen requirements. So oxygen usage and tolerance in microbes. So aerobes, aerobe means that they need air, so they need oxygen is what that means. And so they are a lot of the bacteria there. They need oxygen in their metabolism, and they have the enzymes able to detoxify the toxic oxygen byproducts and stuff like that. And if it cannot survive without oxygen, then it would be an obligate aerobe, so it has to have oxygen to grow. So most fungi, protozoa, and many bacteria, such as Bacillus species and Mycobacterium tuberculosis, are obligate aerobes or aerobes here. So if you put these bacteria in a tube of broth here, this is like they can move around and all the way up through in there. They will grow up at the surface where the air is. Okay. Microaerophiles do not grow at a normal atmospheric concentration of oxygen, but require a small amount of oxygen in its metabolism. So they will grow a little bit below the surface where the oxygen level, there's still some oxygen, but it's lower. Okay. So organisms that live in soil or water or in mammalian hosts are and not directly exposed to atmosphere, such as Helicobacter pylori, which gives us our ulcers, stomach ulcers and stuff, and Borrelia burgdorferi, which causes one of the tick-borne diseases. I believe it's Lyme. Facultative anaerobes. So these guys, they do not require oxygen for metabolism, but they can use it if it's present, but they can also grow in anaerobic conditions. So oxygen, no oxygen is fine. They can adapt, but oxygen is not toxic to them. So that's a big distinction. So many gram-negative intestinal bacteria in staph are facultative anaerobes. So they can live with oxygen, without oxygen, they have the enzymes to detoxify. So if you grow them in a broth, they will grow throughout the broth. Here they'll use oxygen, here they won't. So just do anaerobes. Anaerobes, meaning without air, they lack the metabolic enzymes to detoxify the oxygen byproducts and stuff like that. So, example, many of your oral bacteria and some of your intestinal bacteria, so they don't like oxygen. And so if you put them in a growth tube, they'll grow strictly at the bottom of the tube and not close to the top where the oxygen is. And then you have aerotolerant anaerobes. They do not utilize oxygen, but they can survive and grow to a limited extent in its presence. They are not harmed by oxygen, but so um, but they possess alternate mechanisms for detoxifying the superoxides and other toxic products of oxygen metabolism. So they'll grow throughout. And certain lactobacteria, bacilli, and streptococci are some of these right here. Okay, aerotolerant anaerobes. Okay, so what would you think that Neisseria is? Do you think is it aerobic, macroaerophilic, anaerobic, or facultative anaerobic? So make a selection of what you think it is. And for some of these, I'll give you all the answer like after the lesson in via announcements. Okay, so let's talk about partnerships and relationships between different microbes, whether it be between bacteria and fungi, or bacteria, fungi, parasites, it's like protozoans and stuff like that. So a symbiosis is a general term that denotes a situation in which two organisms live together in a close partnership. Okay. And so the symbionts are members of a symbiosis. So there is a partnership here going on in a symbiosis. Okay. There are three main types of symbiosis. There's mutualism. So they're in a win-win relationship. Organisms live in an obligatory but mutually beneficial relationship. So, for example, our gut bacteria would be an example of mutualism. We would be really sick without them. We need them. We need them to help break down our food. We need them to educate our immune system and all that kind of stuff. So without them, we wouldn't live very long, and they don't survive without having us as a host. So they need us as a host to provide them with the food and the stuff that they need to live. So that's mutualism. Commensalism is the partner that's called a commensal receives the benefits, while the other one is neither harmed nor benefited. So one wins, but the other one is just unaffected. So that's commensalism. And then parasitism is a relationship in which the host organism provides a parasitic microbe with nutrients in a habitat, but the parasite usually harms the host to some extent. So, and that's one of the hallmark of parasitism. It makes it different from commensalism is that you have the harming of the host. You have disease that it causes disease or tissue damage and stuff like that to the host. And the host obviously so is detrimental to the host. Host does not benefit, but only the parasite benefits. But if you think about it, a parasitic relationship is kind of dumb because if you cause harm to your host, then eventually you're going to have to go find another host. So there. And then antagonism, it's an association but not a partnership. So there is, for example, an association between a free-living species that arises when members of a community compete. So this can happen a lot in soil and stuff like that. So there's limited resources, and they are competing for the resources there. So antibiosis is the production of inhibitory compounds such as antibiotics into the surrounding environment that will inhibit or destroy the other microbes that are in the same habitat. So this is like warfare, it's like microbe warfare for the nutrients that are available. So they're using little chemical weapons. And that when we discovered that was the first source of antibiotics. So we just observed what was happening in nature, which was again accidentally observed in the lab, and with a mixed types of bacteria and fungi on the plate. And they were like, wait a second, these bacteria can't grow around these guys because these guys have this like zone where they can't go. And what it was is they were secreting antibiotics, which was preventing the growth of the bacteria in a certain zone, allowing the penicillium, the fungi that produce an antibiotic, to have the nutrients in that zone. So The first microbe then has a competitive advantage by increasing the space and nutrients available to it. So that's antagonism. It is very common in the soil, again, where mixed communities have to compete for space and food. All right, so give me an example of symbiosis. We've already mentioned some stuff. Just type an answer in there. And then we're going to move to our next case. So, at the Mount Union Hospital, a 5-year-old white male child in good general health and physical condition was presented at the Saturday walk-in clinic by his mother. He was brought in because he had a fever, was cranky, and had complained of a sore throat for about 24 hours. On physical examination by the attending residents, the patient had a fever of 39.3 Celsius, so he has a fever, and had considerable swelling and drainage of the pharynx and in the conjunctivae. So the eyes, too. His tonsils were enlarged and coated with a white patchy exudate. So here is the white patchy exudate, something that oozes, right? White patchy exudate right there on his tonsil, and they're enlarged, red, swollen, coated there. He had a red throat and swollen anterior cervical lymph nodes. His ears were clear. His chest sounded clear, and he had no additional remarkable findings on his routine examination. Okay, so the first poll is going to be, what do you think is wrong with him? So is it diphtheria, influenza, RSV, or strep? Incidentally, all of these affect the throat. So figure out which one you think it is and answer it. And one thing I can tell you is this is the gram stain of that organism. So pay attention to it because you're going to have to type it in. But you see it is purple, therefore it is gram positive. The shape, you can see these little dots, all little circles, right? Little rounds, so therefore they are cocci, right there. Cocci, you can really see it here. And they form chains, like chains of pearls. Okay, so cocci in chains would be streptococci. So now type the shape, arrangement, and gram stain on this bacteria. Okay. So what was affecting the child was Streptococcus. What is really, really remarkable about the strep really is the white patches. The white patches on the red swollen tonsils is totally indicative of strep or strep throat. So Streptococcus, when it grows on the blood agar plate, which can play on the blood agar plate, if it's the strep throat, is a beta hemolytic. So beta hemolytic means that it will eat all of the red cells around a colony into a zone that's completely clear. Okay, that's the beta hemolytic one. So strep pyogenes, which is your group A strep throat, and strep A galacticae, which is a group B strep, can do this. If a strep is gamma hemolytic, that means there's no hemolysis around, then it's probably an enterococcus, which is in the strep family of streps. So we have enterococcus faecalis is one of them. And if it's alpha hemolytic, you'll see this green, kind of a greenish zone around here. So you'll see partial hemolysis. And so strep pneumo and strep viridans are both alpha hemolytic strep. So this gives you, pneumonia, obviously. So the infections that can be caused by strep pyogenes, so group A strep, the same strep that causes strep throat, so the diagnosis here for all little patients. So superficial diseases, it can cause pharyngitis, but skin and soft tissues infections, so it can cause wound infections that might mimic a staph infection. Erysipelas, impetigo, vaginitis, and postpartum infections can all be caused by group A strep. But then if it gets really deep in, which is kind of scary, especially if it's a wound, skin and soft tissue infection, it gets really deep, you can get bacteremia, so bacteria in the bloodstream. But then you can also get this necrotizing fasciitis, which is really nasty, where you just have death of the fascia and tissue and just rotting away of stuff. It's really nasty. You could get deep soft tissue infections, cellulitis, myositis, puerperal sepsis, pericarditis, meningitis, pneumonia, and septic arthritis. And then this guy produces also some toxins. So you have toxin-mediated diseases. So as it grows, causing that skin and soft tissue infection or that pharyngitis and all that kind of stuff, the toxins that it produces can cause scarlatina or toxic shock-like syndrome. And so toxic shock syndrome is associated with tampon use and stuff like that. So if you had vaginitis and tampon use, like you can get toxic shock syndromes, and scarlatina, scarlet fever, which you can get with pharyngitis. Okay, and so the last way it can cause disease is immunologically mediated. So your body's reaction to fighting that bacteria, the group A strep, the immune system reaction kind of runs away with itself, and you can get rheumatic fever and post-streptococcal glomerulonephritis. So rheumatic fever can affect the heart, and also can cause arthritis and rheumatic factor to be there, and then you can get reactive arthritis also, but rheumatic fever can affect the heart too. And then this post-streptococcal glomerulonephritis can cause basically acute kidney failure in patients just post having strep throat. So this is a pretty nasty little bug here. And we're seeing more and more resistant group A strep. So which of these is a potential complication of this bacterium? So we have arthritis, impetigo, necrotizing fasciitis, cellulitis, pneumonia, rheumatic fever, toxic shock syndrome, and H, all of these. So select your answer. And so some of the virulence factors, so we talked about those with staph. We're going to bring this back up here with strep. So again, it has a cell membrane, a peptidoglycan layer because it's gram positive. And then it has a capsule and has fimbriae. So remember, the fimbriae allows it to stick, so it can stick to your throat really, really well. The capsule has hyaluronate, so it's really good to evade the immune system. And the capsule also has protein F and protein M and lipoteichoic acid. So protein F allows it for adhesion, also to stick along with the fimbriae. The M protein is evasion of the immune system, so it keeps it from being destroyed by the immune system. And then lipoteichoic acid is a toxin that causes some of the like toxic shock syndromes and stuff like that. So stuff that facilitate it invading your body is it can form streptokinase and hyaluronidase. So both of these there's enzymes that can break stuff, tissue and stuff down, so it can burrow its way into your system. What allows it to evade the immune system is streptolysin O and S. Streptodornase, C5a peptidase, and streptococcal chemokine protease. So these are like basically counter defenses of the strep against your own immune system. And then it also, as a toxin, makes streptococcal pyrogenic exotoxins, which cause you to run fever and stuff. All right, so a little bit more on strep. So strep throat is caused by strep pyogenes, which is group A strep. So you get pharyngitis, you can get scarlet fever, pyoderma, erysipelas, cellulitis, necrotizing fasciitis, streptococcal toxic shock syndrome, bacteremia, rheumatic fever, and glomerulonephritis. Strep agalacticase group B strep that lives in the vagina, and if a mom is group B strep positive and she's not treated, she could spread it to the newborn through vaginal delivery, and so you get neonatal infections of meningitis, pneumonia, and bacteremia. For the women, you can also get urinary tract infection, amnionitis if she's pregnant, so inflammation of the amniotic fluid. Endometritis, wound infections, pneumonia, and bacteremia can all be caused by group B strep. Other beta hemolytic streps can cause pharyngitis, abscess formation, and bacteremia. The viridans group strep can cause bacteremia, endocarditis, abscess formation, and dental caries. So, cavities is what I was thinking. That's on your teeth. And strep pneumoniae can cause pneumonia, meningitis, sinusitis, and otitis media, or ear infections, and bacteremia. Bacteremia is bacteria growing in the blood. So there's all the different varieties of strep that are pertinent to human infections. And let's talk a little bit about bacterial cell divisions. You have a young bacterial cell here. This is represented like a gram-negative, but you have its, all its little components, and if it wants to divide and make more, first it needs to replicate all its chromosomes, and it divides up the ribosomes, replicate ribosomes, and all that. So we double up everything. And then there's a protein, and it grows in size also, so it gets longer. And then there's a protein band that forms here in between, and then that closes up and becomes, and it grows wall and cell membrane and all that, and then it detaches, and then you have two individual bacteria. Now, if it's like a strep bacteria, they kind of stay attached, and that's how you get those long chains. They kind of stay slightly attached to each other. You get identical genetic material, and these two organisms are identical to this one right here. So this is the process of binary fission or bacterial fission. It's basically everything gets copied, everything gets duplicated, the cell divides in two, and now you have two organisms. So as a review question from biology and anatomy, is bacterial or binary fission, is it meiosis or mitosis? Answer that. This process of one cell making two accounts for what we call the logarithmic growth curve of bacteria. And so what happens is, over time, bacteria automatically, they do that. They grow and divide, grow and divide, grow and divide. And so from one bacteria, you get 2. From 2, you get 4. From 4, you get 8. From 8, you get 16 and get 32. So there's doubling with every cycle. And this is the same amount interval of time. So this is what we call their doubling time. And so if their doubling time was, let's say, 30 minutes, then you have one bacteria. 30 minutes later you have 2, 30 minutes later you have 4, 30 minutes later you have 8, and so you have every 30 minutes you double, and in 24 hours you can actually have visible colonies on a petri dish, because you have, with this doubling, you quickly reach, in less than a day, from just one organism you can reach 1 million organisms, which are then visible on an agar plate. Okay. And why is this relevant? So this has to do also with, we know the doubling time on average of a lot of bacteria and stuff like that, but there are some that are really slow growing. And so if their doubling time is, let's say, daily, like they only from going from 1 to 2 is an entire day, then it would take a month to get the growth that you could see on a plate. And so that would be an example, for example, for TB. And that's why if you send cultures out for AFB, which is acid fast bacteria, which would be tuberculosis, it's going to take a month for those cultures to come back, because it's just a slow grower. Okay, so on average, in the microbiology lab, usually you can see, you can get visible growth at 24 hours. But we will definitely check the plates at 48 in case something is slow growing. And we always hold the plates 5 days. And if there's no growth after 5 days for your average bacteria and stuff that are pathogenic, then we can pretty confidently say that there was no bacteria present in that culture. Okay, so which one of these will show colonies on the plate the fastest? The bacteria that doubles every, and then you have 20 minutes, 30 minutes, hour, or day. Which one shows up the fastest as colonies on the plate? And so part of this growing requirement in a culture, if you have to think about it, a culture is a closed system. So whether you put it in a broth or on a plate, there is a limited amount of nutrients and space and all of that. So what happens is when you first plate a culture or inoculate a culture into a broth, there is a little bit of a lag phase, which is an adaptation phase of the bacteria to its new environment. Okay, takes it a little bit and a few hours, and it adapts to its environment, and you start getting some growth and some doubling and stuff like that. And so as long as there's plenty of nutrients and there's plenty of oxygen or whatever the requirements is available, you're going to get exponential growth. Okay. And you'll get many bacteria cell life. So we usually are working in this kind of window right here. We want it to see nice and growing bacteria. And then you have the stationary phase. So in the stationary phase, so this is about a day in a set thing, you start having some bacteria that are dying off. You start have a depletion of some of the nutrients and all that, so there's not as many resources. And so you got about as much, you have some dying ones, but they don't outnumber the living ones here. So this is kind of stationary hold. Then after usually 36 hours and stuff, we have the death phase, where there are some cells that remain viable, but there's more dead cells in the culture than there are live cells, and a lot of the nutrients are just depleted. Okay. And so that's, let me go back to this for a second. So that would allow some time. So if we grow something out and then we need to work with it some more, what we'll do is, if it's been 24 hours or whatever, we may subculture it, plate it, move it to a fresh plate and start back over here and let it grow again. And so you can maintain a culture stable for, by just replating them every day or two, every other day, to remain viable. You can also freeze them at minus 70 degrees and suspend, and then they'll just stay good for a long time. Okay, so let's talk about our next to the last slide, analyzing population sizes of bacteria without culturing them out on a plate. Okay, so that's possibilities. So if you just had them in a clear broth, a clear nutrient solution becomes turbid or cloudy as microbes grow in it, and you can measure the level of turbidity and cloudiness and correlate that to a general population size. Okay, that's one way you can do it. And there's cell counting. So it's on the same principle that we use to count red cells and white cells and stuff, but with machines. So direct cell count is actually measured microscopically. It's actually looking at a drop of fluid on a microscope slab that has a grid, and you actually are counting the number of cells. This is okay for something that doesn't have a lot and where the microbe is rather big, like a fungus or something like that. If it's small or it's probably better to do turbidity, but the culture counter is the way we use to count red cells and white cells and stuff like that. And it basically electronically scans fluid as it passes through a really tiny pipette. So think a really tiny tube where only, like, let's say, one bacteria at a time can pass through. And then every time it passes through and it breaks an electric field, the counter counts as one. And you can count like millions of stuff really, really fast that way. A flow cytometer works similarly to the culture counter, but it can also measure cell size and differentiate between live and dead cells, and it uses more like lasers and other technology. So it's beefed up culture counter basically with more options on it. And then you have genetic probing. And that one uses real-time PCR to quantify bacteria and other organisms present in environmental or tissue samples. We're gonna have a whole chapter on the genetics too. So you can use PCR amplification and stuff like that in genetics to ID and analyze population size and ID bacteria. So anyway, we'll have a chapter about that towards the end of the semester. Okay, so your open-ended question is, of course, do you have any questions? And if you have any questions, pop them in there. And that is part 2 of your lesson on bacteria.